Following the cessation of the Balkan conflict in the late 1990s, more than 40,000 people were missing in former Yugoslavia, including 8,100 people, the traces of whom were lost during the fall of the Bosnian enclave of Srebrenica in early July 1995. Scientists from the International Commission on Missing Persons, or the ICMP, have been working in Bosnia-Herzegovina for well over a decade to identify the victims of war, finally giving certainty to the families about the fate of their loved ones. The only means to achieve this are genetic fingerprints from the victims' bones.
DNA is really the only mode for identification for the majority of these identifications. So DNA is actually giving a name back to those missing persons, where with other modalities, traditional means of identification could not be applied here in this region.
But this task is anything but easy. The remains of the war victims were often scattered among several sites and hidden in various mass grave sites.
Here in Bosnia, we're trying to solve one of the largest forensic puzzles in the world. None of these identifications could be done without the use of DNA. Finding the graves and exhuming the bones and examining the remains is only the first step in the entire process.
However, DNA is subject to a natural decomposition process, more so as the bones have been exposed to the effects of weather for years and are often cross-contaminated by foreign matter. These factors can influence the validity of subsequent findings. Therefore, all contaminations are carefully removed from the bone samples before DNA can be extracted for the subsequent analysis. When the ICMP began its work in Bosnia in the mid-1990s, there were no DNA technologies available that satisfied the high requirements of the scientists. The breakthrough didn't come until 1998, thanks to a technology that QIAGEN developed in Germany, and originally for the processing of blood samples. The main challenge when extracting DNA from bones is that it's usually only present in small amounts and often highly damaged. That means you have to deal with very short fragments, which have often been chemically modified.
QIAGEN's played a very special role with the ICMP, not only in just collaborations, but in the technology also. It's been proven since almost the beginning of ICMP, especially on these highly degraded samples, that the QIAGEN products have worked quite well in extracting as much DNA as possible and giving us a high yield from these very challenged samples.
Thereby, the technology has been consistently refined. The current generation is a miniaturized and automated method, which scientists are hoping will considerably speed up their work. Using QIAGEN technology in the former Yugoslavia alone, the ICMP has analyzed well over 30,000 bone fragments to date, and shed light on the fate of over 15,000 victims.
Yes, you will. Have you seen this case file?
No?
No, it's a nice case file. See, it has all of this information in it.
At the end, after we finalized the identification, after actually they even buried the human remains, family members, some kind of try to find their peace with their lives, because then they have a place to go on.
In the future, ICMP scientists hope that their work could even contribute to active peacekeeping to prevent crimes against humanity from happening at all.
Hopefully we'll actually have an impact on the greater world in that people will realize that these crimes, or what they're committing, will not be able to be hidden. They can't be concealed. These types of DNA technologies can actually tell the truth about what had happened.